Okay, good morning, friends. Yesterday we discussed about various fungal diseases, and in 5 minutes we will have a recapitulation of what we learned yesterday. You know, diseases, we confine ourselves mostly to the diseases caused by fungi, bacteria, viruses, mycoplasma, and then we'll also discuss about various pests. Pests to cause a lot of damage to the standing crops and also the storage crops. So yesterday we discussed about you know the diseases of fungi mostly on important food crops such as paddy, wheat, maize, bajra. So quickly I'll give you what we learned yesterday. We discussed about paddy. Paddy, the staple food for most of the more than 50% of the world population, and paddy suffers from three important diseases caused by fungi. One is blast of paddy, or which is caused by Magnaportha vorizae, otherwise called Pyricularia vorizae, and the other is brown spot of rice, caused by Helminthosporium vorizae, or otherwise called Drechsleria vorizae. The third is Bacane disease. Bacane in Japan, it means what we call foolish seedling. That is, a plant shows excessive growth. The seedling shows excessive growth, and because of this excessive growth, plant will not survive, and it will not, give either panicles or fully filled seeds. So generally, seedlings die. This is what we call a Bacane, and this is caused by Fusarium fusicorum, otherwise called Gibberella fusicori. The excessive growth of this fungus is because of the release of a phytohormone called gibberellin that induces cell expansion. Because of that, plant grows excessively. So these three diseases, that is blast of rice, or what we call brown leaf spot of rice, and Bacane disease, cause a lot of damage to the standing crops, and the extent of damage would be from 70 to 90%. Because we have already learned, just for recapitulation, I am telling this. Then we also discussed about wheat. Wheat is the other staple food for more than most of the North Indians. As far as India is concerned, most of the North Indians take wheat as the staple food, and wheat too suffers from two important diseases. Of course, there are a number of diseases are there, but in our syllabus we have only two diseases. One is what we call stem rust. Stem rust. The other is loose smut of wheat. Now, this stem rust is caused by the famous disease, that is what we call a rust. A rust means it produces streaks or spots which are rusty in nature, rust-like. Hence the name rust. And this is caused by the Puccinia graminis critici. I told you it is a heterogeneous macrocyclic fungus. Heterogeneous means it needs two, hosts for completion. Malaria parasite. So it needs two hosts. Human being is the primary host and the mosquito is the secondary host. So also, in case of wheat also, the wheat which suffers from rust disease, it needs two important hosts, the wheat plant and the barberry plant. These two, the wheat is the primary host and the barberry is the secondary host. And in all, it produces 5 types of spores. 2 spores are produced on wheat. One is uridospore, the other is the teletospore, and the alternate host, that is barberries on barberries. On the upper side of the barberry leaf, the pycniospores are produced, or the pycnia. Pycnium is the and receptive hyphae and spermatangia. These are the sex spores, and the other is echiospores. Now, at the end of the season, generally iridospore on wheat plant turns black. Hence the name black stem rust. So here the spots are not only seen on the leaf lamina, even on the sheath, even on the panicle, even on the glooms, on the stem. You will see everywhere the infected, plant. So at the end of the season, this stem, because of the modification, because of the transformation of iridosaurus into teletosaurus, which are black in color. Hence, the stem becomes black. Hence the name black stem rust. And the other is, of course, loose smut. This is caused by what we call Ustilago sigeatum. Ustilago sigeatum. Here what happens, see, in case of rust disease, the leaves, instead of being green, they turn yellowish and brownish, and ultimately they die. So a lot of photosynthetic effect gets affected. So hence the yield gets drastically reduced. Whereas in case of smut, here the ovary or the seeds are replaced by smut spores. So Ustilago sagittum, here it reproduces by black spores. Black sooty spores are produced, which replace the ovary where the seeds are produced. The entire head turns into black sooty structure, hence the name loose smut. This we discussed, and other things we discussed yesterday include what we call sorghum smut. In sorghum also we find what we call smut. Smut means black sooty spores are produced. The teletospores are what we call chlamydospores, which are black in color, hence the name smut. So in case of sorghum, generally three types of diseases on sorghum are found, and these are caused by the genus Sphacilotheca. So Sphacilotheca produces three types of diseases. One is covered smut, the other is what we call loose smut, the other is head smut. So the covered smut is caused by Sphacilotheca sorghum, and the loose smut is caused by Sphacilotheca cruenta, and Sphacilotheca reliana causes what we call head smut. The other is we discussed about the bajra, otherwise called Penicillium typhoidum or the pearl millet. Here, the infected head, it turns the stamens, the stigmas, and the glooms. The infected head turns into leafy structures. Hence the name green ear disease of Bajra. Green ear disease of Bajra. This is caused by Sclerospora graminicola. So here, in place of seeds, all the floral parts turn into green leafy structures. Hence the name green ear disease of Bajra. We also discussed about what we call mildews. The mildews are powdery mildews. Here, powdery mildews we learned, and these powdery mildews, generally they are found on cucurbits, mostly on cucurbita maxima, that is your gourds, what we call snake gourd, or cucumbers, or pumpkins, or bottle gourd, even on leguminous like our green gram, black gram, even on them also, on the leaves, you will see lot number of spores. The spores are whitish in color, and if you observe that, infected leaf, you will be surprised to see that some ash is sprinkled or some powder is sprinkled. So hence the name powdery mildew. So here also a lot of damage is done because of these powdery mildews, and powdery mildews are caused by what we call, Erysipeus species and Podosphaera xanthi. They cause powdery mildews in case of cucurbits. We also learned about, downy mildews. The downy mildew this is caused by, Plasmopara viticola, and the infect from the lower side of the leaf, you will see, sporangiophores or conidiophores arising tufts, and at the end of the conidiophore, you will see number of conidia. So the conidiophores and the conidia are forming, white cottony structure on the underside of the leaf. Hence the name, the downy mildew. Downy growth. Downy growth is nothing but cotton-like. So it is cotton-like, hence the name downy mildew. Then, of course, up to this we read, and we have two more diseases. Uh, one is uh, one is what we call a leaf spot. Of course, you read about them in your you know uh, earlier first year semesters. So leaf spot of uh, you know, a leaf spot of groundnut. Groundnut is an important oil seal, and in all the groundnut growing areas, the crop suffers from the leaf suffers from what we call leaf spots. The leaf spot is otherwise called ticka disease. Ticka disease of groundnut. This is caused by a fungus called Cercospora. Cercospora has two important species. One is Cercospora personata, the other is Cercospora arachidicola. So both of them cause a lot of damage. So when compared to arachidicola, the loss caused by personata is very large. Generally, they differ. Generally, the leaf spots produced by personata are round in shape and they are surrounded by yellow halo. Whereas in case of arachidicola, the leaves, the spots are very big, irregular, and they are not encircled by halo. And generally in personata, the conidia are 2 to 5 celled, whereas in case of arachidicola, they are very big and the conidia are 4 to 6 or 7 celled. So both of them cause a lot of damage to the groundnut crop. And the last one is wilt of cotton. The wilt of cotton, cotton, we Gossypium herbaceum, what we call cotton. And the cotton is an important crop, and this is a cash crop. Now, the here it causes what we call fusarium species. Fusarium causes what we call wilt disease. Wilt disease, here it is the fungus or the pathogen mainly enters into the conducting tissues, especially the xylem, and it blocks xylem vessels. Hence, the upper parts, upper branches, and the tip of plant body as suffers from minerals and also water. Hence, in due course, the entire plant dies. Hence, it is called wilt. So here, the fusarium reproduces by the production of what we call spores or the conidia. It produces three types of three types of conidia. One is what we call microconidia, macroconidia, and the chlamydospores. So because of them, a lot of damage occurs. The entire plant dries and wilts, and the balls, what we call the kappas, are the balls that are produced are of small size, or even the lint, what we call cotton produced, will be very much reduced. So this is in brief about what we have learned yesterday. Now today, after these bacterial diseases, today we will learn about see, yesterday we discussed about fungal diseases. So today we'll confine ourselves to bacterial, viral, and mycoplasmal diseases. Now to begin with, bacteria. Bacteria are single-celled, prokaryotic, microscopic organisms, and they are found everywhere. So you'll be surprised that bacteria are found everywhere. So they are present in the cold climate, they are found in hot springs, they are found in air, they are found in water, fresh water, marine water, or even, effluents on our body, in our body. So it is not an exaggeration to say that there is no place as such where there are no bacteria. Bacteria are omnipresent. They are present everywhere. They are present everywhere. So now bacteria, bacteria to cause or produce a number of, symptoms. So a number of symptoms are there. So major symptoms are, what is the symptom? So when the pathogen enters, there occurs a kind of a tough competition between the host and the parasite. And, when the host, responds to check the spreading of the pathogen. So in due course, the pathogens upper hand and the sufferer is host. So in response to the entry of the pathogen, it shows certain symptoms. And the symptoms are of various kinds. One is what we call necrosis, and the other is blight, rots, cankers, tumors, galls, wilts are some of the important symptoms of bacterial disease. So necrosis, whenever you know get the necrotic spots, means necrosis is nothing but dead cells. So with the entry of the pathogen, it induces, it attacks the host cells, and in due course, host cells die. The dead tissue of the host cell forms what we call, brownish or blackish spot with dead tissue, and that what we call necrotic spots. And the other is, for example, blight. Blight is nothing but burnt appearance. The leaf, entire leaf, which is greenish in color, it slowly turns into yellow. Later on, it becomes brownish. And this brown and burnt appearance, what we call, blight disease. And of course, the other is what we call rot. Rots are of two types. The rot could be what we call a dry rot or a soft rot. So soft rots are, slippery. See, for example, this is a common thing, if you buy, vegetables or fruits, etc., if you keep them opened, into 3 or 4 days, you will see they're getting spoiled. If you touch them, they are slippery. So this is what we call rot, soft rot. So this soft rot is caused not only by bacteria, but even fungus also causes this soft rot. So we confine only to, bacterial disease, hence the soft rot. See, generally, what we call Erwinia. Erwinia causes what we call soft rot in case of carrots. So carrots, if you buy carrots, if you keep them in an open area for 3 or 4 days, if the climate is like this, if a damp climate is there, if relative humidity is more, then the tissue, because of with the secretion of certain enzymes, proteolytic and lipolytic enzymes, the membrane and the middle lamella gets dissolved, and it becomes leads to the production of a watery mass oozing. And that watery mass, see, you have to remember the bacterial, spot. From the bacterial spots, you will see certain watery, soft mass oozes out, and it smells bad. So this tells us that that particular disease is caused by a bacteria. So the rots are Erwinia and Bacillus, Pseudomonas, and Clostridium cause rots in case of vegetables and fruits. Now we have what we call tumors or galls. Tumors and galls, generally the tumors and galls are boil-like structures. They are nothing but excessive growth of the cells and tissues of the host. This is induced by the pathogen. So with the increase in cell division, what we call hypertrophy, hyperplasia and hypertrophy, both together increase the size of that particular lesion or particular part. So generally, galls we will see, for example, even in case of maize. In maize also you will see galls and boil-like structures. And if you disturb them, if you crush them, you will see white mass oozes out. White watery mass oozes. This is what we call, tumors or galls. And in addition to that, what we call scabs, scaby. What we call scaby is generally scaby lesions. Generally, you will find in underground, especially on the roots, on the tubers. In tubers, maybe your sweet potato or what we call our potato or even roots, you will see this scaby structure. Even on apples also, this is common. This KB, small rough structure that deforms the healthy tuber, what we call scabs. So scabs is also caused by what we call the bacteria. And the other is wilts. So here bacteria, generally Erwinia, Corynebacterium and Pseudomonas and Xanthomonas, they make an entry into the host. How do the bacteria make an entry into the host? Generally, there are natural openings, on the leaves, you have what we call stomata. Through stomata or through the wounds, they make an entry. They enter into the host and they replicate. Bacteria have the ability to reproduce very fast. So they rate, within 20 minutes, bacteria like, our cholera bacterium, it multiplies. So hence, certain diseases, bacterial diseases spread very fast. So these, bacteria, they multiply with an enormous speed and they enter into, conducting tissues, especially your xylem and phloem, and they, release certain enzymes and with the release of these enzymes, there is what we call clogging. The clogging or the blocking of the conducting tissues takes place and the entire plant body dries. So these are some of the symptoms. This is an example, important question, what are the various symptoms caused by bacteria? Write a short note or an essay on bacterial symptoms. Then important question. Then we have 2-3 diseases, caused by bacteria. One is paddy. In paddy, bacterial blight. Bacterial blight is caused by Xanthomonas oryzae. And, the bacterial blight and the fungal blight. In fungi also we have seen blight disease, Pyricularia oryzae. The Pyricularia oryzae and the bacterial blight differs. Both of them are, host is same, rice. But the spots, if the spots, you will see white or pale yellow streaks or spots on the leaf infected by the bacteria, and as a result of the entry of the bacteria, with the release of various enzymes, the tissue gets, the middle lamella and the membranes get dissolved, and it produces what watery drop-like structures. Watery drop-like structures are seen as if, if you observe certain grasses, in the morning, early morning, you will see water drops. That is what we call guttation. So here also, the infected leaf shows, watery drops, which are hyaline or which are grayish earlierly, but as the time passes, they become, yellowish. So with the appearance of these watery yellowish drops, that denotes that the disease is a bacterial disease caused by Xanthomonas oryzae. So Xanthomonas oryzae is a bacilliform and a gram-negative bacterium. It is monotrichous, as a single flagellum. Generally, the disease, the bacteria are seen, you will see the bacterium, it remains alive in the soil or in the stubules or fallen leaves, or, even your seed also has the pathogen. So hence, to check the spread of the disease, what we have to do, you have to treat the seed with, seed dressing, either dry dressing or, wet dressing before sowing. So how to check, this? The best thing is, the fertilizers, chemical fertilizers have to be given in the splits, especially nitrogen has to be given in 3 splits. And generally you go for what we call crop rotation and do what we call seed dressing. And if you find, see that your water canals, or hedges, etc., do not have any collateral hosts. And generally, go for what we call the best way is go for this paddy blight resistant varieties, especially IR64 is there, and Ajay is there, Sona Masuri, Swarna Radha, and what we call IR20. These are the blast resistant varieties. And if you still, if you instead of, in spite of taking all precautions, if you find, the disease on the crop, generally you go for what we call spraying of certain, bactericidal, such as agromycin or cerasone, neem oil, copper sulfate, or streptocycline, etc. You can spray and check the spread of disease. This is bacterial blast. Then the other, disease caused by a bacterium that is there in our syllabus is citrus canker. I think you know about this. Even we discussed in your practicals also in the first year. So here, citrus canker, what we call the citrus canker. The canker is nothing but what is the canker? So the canker is nothing but the raised structure where you find dead brownish dead tissue, and that is covered by living tissue. So the raised watery structures are called what we call cankers. The cankers are seen on all aerial parts. You will find them on the leaves, on the young stems, or even on the old stems, even on the fruits also. This is a common thing you might have seen in Nimbu. If you buy this our sweet lemon, you will see certain watery structures. If you using either a knife or your nail, if you open that, in that watery structure, in the center you find what we call brownish dead tissue, and that superficially covered by greenish living tissue. So these watery structures are called cankers. So now this canker disease is very, very common in all, these Nimbu growing areas. You find this. So here the thing is, unlike other crops, neem is an orchard, and generally, we keep that orchard for 10 to 15 years. So if your plant gets infected with this bacterium, unless and until you prune it, you cut them, you burn them, it remains. So hence one should be very, very careful while getting the saplings of this, citrus from the uninfected areas, and see that your preparation of is done, and also all precautions are to be taken, and regular checking has to be done. So after harvest, what you have to do? You have to see, you have to prune, you have to cut the infected areas. Your leaves, of course, they fall, but even these cankers are found on the branches, and also on the trunk, main trunk or big branch. So those are to be cut. These cut portions are buried and burnt, so that your next crop will be free from this bacterial infection. So here you will see cork browny structures. I told you, generally you will see living green living tissue over that. So by this canker, say for example, you and me, if you go to a shop, we look for the yellow one, big one, and non-canker, or the fruit that is without any canker. So here, if your orchard, if your plant suffers from this cankers, the fruits produced will be of small size, and they are deformed. Your marketing, marketing quality gets reduced. Farmers suffer. So not only in terms of quantity, even in terms of quality also, the farmers suffer. So we have to be very careful, and generally here this is caused by Xanthomonas oxynopodis citri. So here they like our blast of rice also. Here also it is a gram-negative bacterium. It is a bacilliform bacterium, monotrichous bacterium. So here, what you have to do, so the precautions should be pruning. So pruning is after the harvest. What you have to do during generally in off season. After harvest, then what we have to do? Remove all the diseased foliage, branches, etc. by using scissors, etc. And these scissors and other implements, what you use, um, you have to see them, you have to clean them, you have to disinfect them, or else even there is every possibility for the transmission. If you use contaminated scissors and other sickles, etc., there is every possibility for the entry of the pathogen into the healthy areas. So, and generally get your seedlings from the nursery, which is free from any disease, and the fallen infected leaves are to be collected and they are to be burned. And the other thing is, go for spraying of what we call Boradox mixture and lime sulfur, etc. They are to be sprayed. And the other is, because it is a bacterial disease, your antibiotics can be sprayed, like streptomycin and phonomycin, and even spraying of neem cake found to be very effective in controlling this. This is so we have only two diseases caused by bacteria. One is blast of rice, the other is citrus. Then we move to viral diseases. So viruses are the obligate parasites. They are the obligate parasites, and they are the smallest organisms. Of course, there is a controversy. Some people consider viruses as living, the other considered as non-living. So safely, they are considered as a bridge between living and non-living. So why they are non-living means, if the virus is outside the host, it remains outside for a number of years without undergoing any change. If it happens to get any suitable host, then it gets awakened and it makes an entry. And by using the host machinery, it multiplies. The other living characteristics are its ability to reproduce and its ability to mutate. COVID virus, it underwent 100 times of, mutations. Some mutations are harmful and most of the mutations are very, harmless. So here also, the virus undergoes mutation. And of course, viruses, they cause number of diseases. They cause so we suffer a lot from viruses. Viruses cause number of diseases in human beings, animals, and also in plants. I think I told you the COVID is of course a viral disease, and your common cold is a viral disease. Polio is a viral disease. So we have your measles, mumps, of course. So we have n number of. So the other is what HIV, papilloma virus. These are all viral diseases. Viruses cause number of diseases in human beings, and even they cause number of diseases in plants. We have 3-4 important diseases caused by viruses, and the other like bacteria. Here also the plants produce a number of symptoms. What are the important viral symptoms? This is an important question for you. So various viral symptoms of viral diseases in plants. So the best one is mosaic. So whenever I say mosaic, I think you'll be able to recollect TMV, that is tobacco mosaic virus. It was the first viral disease that was, noticed by Ivanovsky, Weisering, Meyers. A number of people worked a lot on, viral diseases, especially on tobacco mosaic virus. Viruses differ from other organisms in one characteristic feature. So in all our organisms, you find both DNA and RNA. But viruses are the exclusive group where the viruses contain either DNA or the RNA. Based on their, this nucleic acid, viruses are classified as, DNA viruses and RNA viruses. Generally, plant viruses are, RNA viruses, whereas animal viruses are, DNA virus. But it's not. But there are many exceptions. See, for example, COVID is an RNA virus. So tobacco mosaic virus is a RNA virus. And here, the viruses cause what we call mosaic. What do you mean by mosaic? Mosaic is nothing but cheetah, what we call. Say, you might have seen, the ripened, banana. The street vendors call, cheetah banana, what they say. Cheetah means spots, various shades. So the infected leaf shows what we call various varied shades of green and yellow. These varied shades of green and yellow make the leaf variegated. That variegated leaf, what we call mosaic. So mosaic is mosaic diseases is present not only on tobacco, but even cucumber mosaic virus is, cauliflower mosaic is there, even tomato mosaic, where a number of mosaic diseases are there. So in addition to tobacco, many Solanacean members do suffer from this mosaic disease caused by viruses. Now, one is mosaic. The other important symptom is what we call vein clearing disease or vein banding disease. So whenever I say vein clearing, bandy vein clearing disease. You might have seen, bandy, what we call lady's finger. The plant, if it suffers from, viral attack, if you observe the leaf, you will see the veins are like this, palm-like. And now you will see all the veins turning yellowish. Hence, it is called vein clearing disease. And in addition to that, in some other cases, for example, in case of soya bean and in case of papaya, in case of strawberries, etc., you will see vein banding. So instead of yellowish nature, you will see green bands. So hence the name vein banding. Vein clearing and vein banding is the other. And the other is what we call ring spots. Ring spots will be there. And these spots are very common on the leaves. And generally, the healthy leaf will be greenish in color. Now, the infected leaf, shows the spots. These spots are generally yellowish in color. Later on, they turn, what we call a brownish and dark. And the other is necrosis, of course, I told you. So necrosis is nothing but dead tissue, dead cells. So in response to or with the entry of these viruses, the cells get damaged. The damaged cells, dead cells form what we call dark brown or dark spots. They are called necrotic spots. Now the other is wilt. So wilting is nothing but drying. Death of the entire plant, what we call wilt. Now the wilt is because of here also viruses make an entry and they are seen entering into your conducting tissue, that is phloem and xylem, and in response to this, there is a tough fight between the pathogen and the host, and the pathogen blocks the conducting tissue, hence the upper part. Initially, the upper leaves, the upper part of the stem dries and later on it spreads downwards. So ultimately the entire plant dies, what we call wilt. The other is what we call stunting or premature foliage. So when plant suffers from viral diseases, the suffered plant, the growth gets checked. Hence, normal growth gets affected. The leaves will be very small and the plant stature will be very small. The fruits will be very small. So this is what we call stunting. Stunting growth. This not only reduces your quality, but also the quantity gets reduced. The other is distortions. Distortion is nothing but the leaves, fruits, etc. They get deformed. They lose their luster and shiny structure, and they get cracks and folds and deforms. So the deform will be either on the leaf or on the what we call panicle or the inflorescence, or even on the fruits, etc. So distortion will be there, and mostly, and even the vein cells, veins do not expand. So hence the lamina or the green part gets expanded, but because of the non-expansion, non-extension of the veins, you will see folds. Folds and crinkling and rolling of the leaves will take place. And even tumors. Even tumors, of course, even in human beings also we suffer from certain tumors. Tumors are nothing but cancerous growth. Cancer is nothing but uncontrolled growth. So even with the entry of these viruses, they cause what we call tumors and gots, uncontrolled growth. And the last one is what we call flower breakings, especially in case of tulips. Tulips, these are found in Kashmir, of course, in Dal Lake areas, or if you go to Rashtrapati Bhavan in Delhi, there you will find tulip gardens, tulips. Now, these tulips, with the entry of this pathogen, the petals show what varied shades. They add extra attraction to the flower. So this is what we call flower breakings. So these are, symptoms. Now we have disease, one disease is there, the other, that is what we call Tungro disease, rice Tungro. So rice suffers from what we call Tungro disease, and this Tungro is caused by a virus. And there are two types of viruses, what we call, one is rice Tungro bacilliform virus and rice Tungro spherical-form virus. These together cause Tungro disease. What do you mean by Tungro? Tungro in Indonesian language, Tungro means reduced growth. The plants do not show. We have seen in Botany, wherein plant shows excessive growth. Whereas plants suffering from rice, suffering from a Tungro virus, they do not grow. They are very small stature will be there, and they fail to, produce panicles and no flowering and no fruiting, no yield. So Tungro virus cause lot of damage, especially in case of Southeast Asian nations such as your Thailand, Indonesia, and Philippines. Their rice field suffers from, what we call this rice Tungro virus. Now symptoms, generally the plant will be, because I told you reduced growth. Plant shows what we call stunted growth. Stunted growth, the internodes get stunted, and plant becomes what we call bushy-like, and too many tillers will be there, very small, no flowering, and no seed setting will be there. So here you can also see what we call intervener chlorosis will be there, and generally young leaves suffer initially, later on spreads to older leaves. And generally tillers will be very few, and even root system gets affected, and the internodal part gets affected, and the stunting will be very, very severe. So the rice plant will be of this size only, stunted. So this is what we call Tungro virus. So here, the Tungro virus, you have to remember very important thing. See, unlike viruses, generally they spread. Spreading will be generally by human beings or by using various implements, etc. So here, the Tungro virus spreads to healthy parts or the neighborhood because of what we call a green hopper by name Nephotetic virescens. Nephotetic virescens is an insect that sucks sap from the leaves, and when it visits another, so whatever the virus that are there entangled to its mouth parts, they are released in the next visit of the healthy part. So hence what you have to do, so here you have to check the population of insects. So various insecticides have to be sprayed to check the spread of the disease. So generally what we have to do here, generally you do sanitation, that is keep your field very clean, and at that transplantation time also you see that the vector population is very less, and the seed bed has to be free from pathogens. And here the chemical control found to be very effective. Generally Furatox is there, and Furadon is there, and generally carbofuran and cypermethrin, and carbofuran, cypermethrin, and even 2,4-dimer, 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 2 uh, furadon and carbofuran cypermethrin are found to be very, very effective in control of this. Even bendiocarb, these the other insecticide that has been found to be very effective in controlling the green green leaf hopper. That is uh, the main distributor or spreader of the disease, that is nephrotics virescens. Then, of course, tobacco virus, very well known, and tobacco, Nicotiana tabacum, and the common, you know, uh, disease found in tobacco is tobacco mosaic virus. And of course, I already told you it was Ivanowski, later on Beisering, and uh, other people worked on this, Mayers and other people worked on this. And tobacco primarily suffers from the tobacco mosaic virus. And this tobacco mosaic virus is a RNA virus. And generally, if you look at the RNA virus, there are two important parts, what we call the core and the capsid. The core is the nuclear material. Here, the nuclear material is RNA, and the capsid is what we call proteins. So, hence, the viruses are called nucleoprotein particles. The nucleoprotein particles. So, they cause a lot of damage to the tobacco leaves, and generally, infected leaves will be of their size gets reduced, and not only size, but even the quality gets affected. So farmers suffer a lot because of the infestation by tobacco mosaic virus. So here the spreading, generally implements, and even the workers or the BD cigarette and the scissors, etc. While doing, while removing, topping inflorescence, etc. While cutting also, whatever the scissors and etc. implements, scalpels, etc. they use, they have to be disinfected, or else there is every possibility for the spreading of the disease, mainly through human beings, especially through implements. So here, because viruses, there are, you'll be surprised, there are no medicines to control viruses. There are no medicines to control viruses. Even if, there is a saying, if you suffer from what we call, cold, if you get relief in 7 days. Even if you visit a doctor, you get a relief in 1 week. So 1 week, 7 days, same. So there are no medicines that counter the spread of the disease, mainly caused by viruses. So here vectors are to be, and the implements are to be checked. Then last one is mycoplasma. So mycoplasma is another, these are the smallest known organisms. They are the smallest known organisms. They are smaller than your bacteria. Bacteria we consider smallest, but mycoplasma are the smallest known organisms. They differ from bacteria in lacking any cell. Cell wall is absent. So it was Knockout and Raux. Knockout and Raux were the people who first identified the mycoplasma. They were called PPLOs, pleuropneumonia-like organisms. So Knockout and Raux, first of all, they identified them in 1898. So the mycoplasma are known to cause number of diseases. So mycoplasma, they are known to cause number of diseases, and the important diseases caused by them are what we call there are three diseases that are there in our cell. One is little leaf of brinjal is there, and we have grassy shoot of sugarcane is there. So we have what we call phyllodes. Phyllodes is an important disease. Generally, you find this phyllodes in Sesamum indicum. In Sesamum species, the floral parts, that is stamens, stigmas, the other floral part, calyx and corolla, they turn leafy structures. So hence inflorescence becomes leaf-like, hence the name Phyllodes. And the other disease is spike disease in sandalwood, Santalum album. Here, the infected plant, the infected plant, the leaves become very small and the nodes become very small, and it changes into a spike-like structure. Hence, the called spike disease of, sandalwood. And we have the important thing is in syllabus, what we have is little leaf of brinjal. Little leaf, as the name tells you, leaf becomes very, very small. Brinjal leaves are very big. They're spiny. But infected leaves become non-spinous and the leaves become very small. And with the reduction in the leaf, even the fruits, eggplant, what we call, the brinjal, the fruit becomes very, very small. And it loses its, watery content. The fruit is very, very hard and it is not fit for consumption. Marketing value is lost. So the quality gets lost and even the quantity gets lost. The plants become very short and the leaves become very small, and the fruits become very small. Hence, it is called what we call little leaf of brinjal. So little leaf of brinjal is caused by the mycoplasma. So the mycoplasma, I told you have to remember, they lack cell wall. Hence here, the antibiotics do not work on mycoplasmal disease because most of the antibiotics work on, they check the cell wall formation. Because they don't have any cell wall, they are ineffective in controlling, antibiotics are ineffective in controlling the mycoplasmal diseases. So grassy shoot of sugarcane is the other disease. The sugarcane, stem, it is unbranched, stem, unbranched with nodes and internodes. If the sugarcane, Saccharum officinarum, if it suffers from what we call mycoplasma, it shows, from the base of the sugarcane, you will see too many tillers. Many tillers will come and the tillers are of, grass-like with nodes and internodes. They don't have any juice and marketing ability gets lost. So a lot of damage will be done. So instead of, juicy and stout sugarcane, you will see small grassy structures, a bunch of grasses from the base of the stem. So hence it is called grassy shoot of sugarcane. So these are the important, diseases of what we call the bacteria, viruses, and mycoplasma. Then quickly, we'll discuss about, the pests, cotton pests. So cotton is very, very important for you in examination. They'll ask you about the importance of cotton or the important species of cotton, important varieties of cotton. Cotton, Gossypium herbaceae, member of Malvaceae. There are 4 important, Gossypium species that are cultivated in India and elsewhere. China ranks first and India ranks second, and followed by, United States and Brazil and Russia. So in India, we grow 4 important varieties. 2 are what we call Gossypium herbaceum and Gossypium arboreum. These 2 are called what we old world cottons, and they are diploid. So their 2N number is 26. Whereas new world cottons, what we call Gossypium hirsutum and Gossypium barbadens. These are called new world cottons or upland cotton or American cotton. They are tetraploids, where the 2N is 52. Cotton, now we are proud to say that Hindus are considered as the first, people who had the knowledge of growing cotton and also, weaving and making threads and clothing, etc. So, number of excavations, especially in the Sindh province, which now starting from your, Afghanistan and Pakistan and Western Punjab area, where Indus Valley civilization existed, Harappa and Mohenjo-daro. In those excavations, people found that this cotton and threads, etc., when they were tested with, carbon dating, they are found to be 6,000 years old. So cotton, important cash crop, it's an important cash crop. And most of the, after what we call the cotton and the sugarcane, these are the two important cash crops on which lakhs of people, depend. Farmers grow them, and the ginning, laborers work, and the ginning will be there, then clothing will be there, and then sellers will be there, tailors will be there. So it's a big industry. So in India, generally, Gujarat ranks first, followed by Maharashtra. There you will see lot number of cotton mills, fabric mills and cotton mills are located because there we have block cotton soils, which are very suitable for cotton growing. Now we have, cotton, cotton suffers from two important pests. One is what we call the first, disease, pest is tobacco cutworm. Tobacco cutworm doesn't anything related to tobacco. So the Spodoptera litura, this is the, leaf sucker. It sucks sap from the leaves. Now, these adult ones, they have, two pairs of wings. The forewings are brown and with black, whitish spots, and the hind wings are white and with brown spots, And these adult one, it lays hundreds and thousands of eggs on the lower side of the leaf, and these eggs hatch into young ones, that is larvae.